Okay, let's get started. Maybe someone will join in the process. Good. Okay, so I hope you missed our lectures on biochemistry because today we have one more, and this lecture is about, I guess, maybe one of the most marvelous biochemical processes in the whole biosphere. That's photosynthesis. So today we are going into certain detail of the photosynthesis, and well, that will be for two lectures. So today we start considering that, and tomorrow I guess we will more or less finish our consideration of the photosynthesis. Good. So first of all, who can photosynthesize, and what are these organisms, and basically what are their properties? Uh, First of all, the process of the photosynthesis can be divided into two large branches. So the first branch, that's so-called oxygenic photosynthesis. That's the process that we will be most interested in. Well, I guess you can guess the reasons why. Because we all breathe oxygen, and of course we are interested in the process that basically produces that oxygen. It is performed by so-called oxygenic photosynthesizers. These are a large number of organisms, starting from plants, of course, that we see every day, and green algae, and red algae, and cyanobacteria, and even some more organisms. Well, I can say you that there is even a sea slug, I guess, from the family of Elysia or something like that, that can hijack the chloroplasts from certain green algae and uses that chloroplast to photosynthesize. So we even know at least a few species of animals, well, namely these sort of sea slugs, that can perform the photosynthesis. Of course, they need to get these chloroplasts from somewhere. They are eating the algae and they are basically hijacking their machinery for photosynthesis. But yeah, that's pretty possible even this way. And you see that it's performed with the pigment so-called chlorophyll A. We will get into the details as we move along the lecture, and we will see how do we get the product, namely glucose, and so on. Before we get into that photosynthesis, just as some sort of note for you, there is of course the second branch, so-called anoxygenic photosynthesis, performed by anoxygenic photosynthesizers. They do not produce any oxygen, well, because at the terminal electron acceptor, at the donor, they do not use water, but they use different substances like H2S or some iron, ions, or organic acids, and so on. They use bacteria chlorophylls to absorb, for example, infrared light, and there are many such organisms as well. So some sort of purple sulfur bacteria, or non-sulfur bacteria but still purple, or green sulfur bacteria, and so on. So you can check this list later. Good. Here are some photos of today's heroes of the day. So that's a microscope image of Cylindrospermum, a filamentous genus of cyanobacteria, performs oxygenic photosynthesis. Here is a Prochlorococcus, an influential marine cyanobacterium that produces much of the world's oxygen, well, approximately 10 to 20%. It's pretty interesting thing that phytoplankton in the ocean produces something around 50% of the world's oxygen. So we are used to think that trees are main producers of the oxygen, but in the ocean there is produced no less than 50% of the oxygen, maybe even more. I don't know. So I should check some articles to review the numbers, but I'm certain that it's not less than 50%. Well, here are some types of non-oxygenic photosynthesizers, so you can see photos of these nice little living organisms. So chromatium, purple sulfur bacteria, they oxidize sulfide to produce sulfur, which is deposited in intracellular granules of the cytoplasm. So if you look closely, you may, I guess, see that sort of granules. Rhodophyllum rubrum, the photosynthesis of these Rhodophyllum rubrum differs from that of plants, as it possesses non-chlorophyll A but bacteria chlorophylls. And here is Heliophyllum fasciatum. Well, this is a phase contrast micrograph of these cells. So here we have a few of them. Here are, once again, different algae, still photosynthesizing. Here are red algae, brown algae, green algae, and some sort of diatoms. Well, I just want to show you that they look pretty different, so there are pretty different species, but they all photosynthesize, and of course we are, in some sense, interested in what are the processes that are basically underlying the photosynthesis. Here is a small comparison table that contains, first of all, the main chlorophylls. Of course, we will consider chlorophyll molecule today because it's a really important part of the photosynthesis machinery. But why are they so different in their color? Because they contain other chemicals. For example, here are some sort of key carotenoids and phycobilins that they may contain and that basically define their color. So here you see the color, here you see some sort of examples and types of organisms that photosynthesize. Of course, we'll be more, I guess, interested in different plants, and I will focus on the plants that we know, but it's just some sort of reminder for you. Some more reminder that the total number of plant species is approximately 360,000. Also, some estimates there may be as many as 2 million diatom species alone, most undiscovered and unnamed. So, well, if you want to discover some new species, that may be a good hint for you. So maybe you need to consider some sort of algae and you will find some new species that you can name as you would like. So here's some info, and I will not stop on that. It's just some sort of reference table for you. A little bit of reminder about land plants. Well, because I'll mostly focus on the land plants as we go. So here is some sort of evolutionary tree that we may draw from for the land plants. And from this evolutionary tree, you see that they originated something like 475 million years ago. So that's the around some sort of beginning of Paleozoic era. And they survived, of course, up to the modern days. Here you see different types of these plants. Some have retained more or less some primitive characteristics. Some other have developed something more and something more modern, let's say. So, for example, in Cenozoic era, you see that we have at last we have flowers and fruits. So that's pretty typical for the plants that we know, or at least the plants that we think about when I tell you the word plant. Okay, what about that plants that we see daily and that we think about, let's say, when we talk about photosynthesis? So the first that comes to our mind. So despite the fact that many organisms photosynthesize, when we get to photosynthesis, probably the first thing that comes to our mind is some sort of a tree. And here is an example of a tree and the different, let's say, scale that compares this tree and different parts of this tree with different scales. So you see, this is some sort of palm. It's quite high, something around 10 meters. But when it gets to a leaf, leaf is just 0.1, 0.2 millimeters thick. We'll be interested in chloroplasts, so I'll get to that. That's something on scale 5 to 8 micrometer. So I guess you remember this mu, that means micro. And we can get even some smaller structures like plasmodesmata, that's 50 to 60 nanometer in diameter. Here is for comparison DNA, just 2 nanometer, and biological membrane that is 5 to 7 nanometers thick. So that's some sort of scales for you how that looks like. And here's pollen, 10 to 20 micrometer. Good. Here is some example of this sort of plant. So here you see some stem from a lilac bush. Here you see some nodes, and you see, of course, leaves. And here are some names of the parts of the plant. So here you see the root system, the shoot system, and some sort of other components, let's say. So here we have leaves. We will be mostly interested in leaves because, of course, the photosynthesis is mostly performed in leaves. And here you see the blade, the petiole, and the axillary bud, and we will consider some parts of that during this lecture. Good. Here is a typical leaf. So that's a typical foliage leaf, macrophyll from a rose plant. So nothing really special, just some sort of photo. What do we see? We see the adaxial surface, so here it is. We see the abaxial surface, and here we see some sort of cross section. So this is a cross section with adaxial towards the top. So here was the adaxial surface, so that's towards the the top. So it's here. So this side towards the bottom, so it's here. And here are some more examples of leaves, just to give you some general idea that they may have a very different form, and here, but they all at least in this picture they all photosynthesize, because in the next slide I guess I will show you a few leaves that do not, but they are still some sort of forms of leaves, and we'll get to that in a slide. So you can just look at the southern wood fern and some Swiss pine and banana. So here how banana looks like. The interesting thing that the entire trunk of the banana, well, this part is basically composed of tightly wrapped leaf sheets. So it's probably we can't call it a tree in some sort of our classical meaning, because this part, the stem, is composed well from leaves. And here are some sort of modified leaves. Some of them do not perform photosynthesis. So here are some examples like cataphyls or bud scales that surround the terminal buds on lilac. Here we have clematis tendrils. So these tendrils are modified leaves as well. Insect trapping picture, here it is. That's modified leaf as well. You have here a golden barrel cactus. and the stem is covered with spines, and these spines are basically modified leaves. And even onion bulb is basically stem and leaves, and you see that bulb scales are basically modified leaves. They do not perform any photosynthesis because that's underground and they don't see any light, so there's no need. Okay, we have seen many different leaves. Now let's get a little bit closer, so let's zoom in. Here is a cross section of a leaf of the Colorado cinquefoil, and here you see on the left how that leaf looks like. Here it is. And here on the right we have a cross section. Maybe we don't need it so badly, but at some point, I guess in the next lecture, we will get to the difference between C3 and C4 photosynthesis pathways, and there will be certain anatomical features that you may notice when you look at the cross section of the leaf of the C3 photosynthesizing plant and C4 photosynthesizing plant. There is some sort of so-called Kranz anatomy. Well, I'll show you that as we get to it. Here, what I want you to see, here are certain features. First of all, this part is called mesophyll. These are the cells that mostly perform photosynthesis. You see this part that's so-called vascular bundle, and when we get to that C4 photosynthesis, we'll be talking about some cells in the vicinity of this vascular bundle. So we will need that at some point. Here you see certain anatomical feature like that you can divide your mesophyll into two parts, so-called palisade mesophyll. You see that cells, they are mostly arranged in some sort of rows, so like one by one by one by one. And down here you see the part that is called spongy mesophyll, and you see that here cells are a little bit more disordered. So here at the top we have some sort of order. They are like aligned in some rows, while at the bottom they are more disordered, and that's why there are much larger gaps between these cells, and that looks like a sponge, so it's called a spongy mesophyll. Good. What's this? And this is a sort of video with an experiment that I wanted to show you, and probably probably we will explain some parts of it. Oh.
Water, but for this experiment, we need them to sink. We'll put our leaves under a vacuum so that the water infiltrates them and pushes the gas out, causing them to sink. By adding baking soda to the water, we provide a source of carbon dioxide for the leaves to photosynthesize, and as they produce oxygen, they'll start to float again. We'll time how fast the flotation takes and use that as an estimate of photosynthetic rate. I'll go through that in much more detail as I walk you through the experimental setup. Let's get started. We'll need baking soda, aka sodium bicarbonate, diluted dish soap, about 5 milliliters of soap into 215 milliliters of water, a pipette, 10 ml syringes without needles, living leaves, today I'm using baby spinach from the grocery store, a hole punch, clear cups or beakers and a way to label them, aluminum foil, a timer, and a light source. I'm using a grow light, but you can also use a desk lamp. Just make sure the bulb is super bright, something like 100 watts. And if you want to compare results across multiple experimental setups, the light source must be the same wattage across all of them. First thing I need to do is prepare a 0.2% solution of sodium bicarbonate in water, so when it dissolves, there'll be plenty of carbon dioxide in there for our leaves to photosynthesize. I'm adding 0.6 grams of baking soda to this bottle of water and bringing it up to 300 mils. That solution goes into one of my cups, which I'll label with CO2. I'll add plain water to another cup and label that one without CO2. This cup will serve as our control, and from now on, I'll duplicate every...
Okay, here's a tricky question for you. So why are we adding baking soda to the water? Who can tell me? Neutralization? No, there's nothing to neutralize. It's just clean water, so we have no acids there, and there's basically nothing to neutralize. Please recall, what is the chemical formula for the baking soda? In some sense, yes, sodium HCO3. So here you see that's a salt. A salt of what? A weak acid and a strong base. So you have sodium HCO3. When you get that into the water, you will get what? You get an ion of sodium and no, not so easy. Yes, you're close. You're close to the right answer, but almost there. So yes, that's the correct acid, but when sodium and this, when soda dissociates, you get basically an ion of sodium, so sodium plus, and HCO3 minus. So you see the dissociation. So everything we learned about dissociation, that plays a role here. Why this is useful to get these sort of ions in water? That's because we remember that CO2 can react with the water, effectively creating I guess I need to put arrows here and there that may dissociate. I hope you remember this, because we have already considered, when we talked about pH, we have already considered the hemoglobin. And when considering hemoglobin, I told you about this sort of reaction. So you can put a lot of CO2 into the water because of these reaction. That's probably the reason then when you have this sort of freezy water, you get it just with CO2. You can't use other gases. There are many gases that are not toxic or something like that, so why don't we use some sort of helium for that, or I don't know, some other gases? But the reason is pretty simple. CO2 reacts with water, so that's why you can put so much CO2 into the water that you can get all that different drinks. And you see that this reaction creates carbonic acid, and this carbonic acid may dissociate. And basically putting baking soda into the water, you get that ions of the carbonic acid. So that's what we have. And of course, if we will get some sort of a proton, and you have some decent amount of them even in the clear water. So you remember that water undergoes that process of rejecting a proton, and you can get back H2CO3, and you can get back at some point CO2 and water. Good. So this is basically the source of the CO2. So all reactions we perform with CO2, we mostly perform in this form. Good.
Step of the experiment across these cups. Next, I'll use a pipette to add a drop of the soap solution to each cup. Leaves are covered in a waxy cuticle, soapy that causes suds though.
Yum.
If I see suds, I need to dilute what's in the cup with more bicarbonate solution or water. Now it's time to prepare our leaves. Whole leaves are too big and irregularly shaped, so I'm actually going to make little uniform leaf discs with a hole puncher, 10 per cup. This is pretty straightforward, but just be sure to avoid big veins because they're harder to infiltrate with the vacuum. I'll be creating the vacuum using the syringes. This is the trickiest part of the experimental setup. I've labeled the syringes the same as my cups. First, I'll remove the plungers and load the leaf discs into the barrels. Then gently replace the plungers so that only a tiny volume of air remains. Next, I'll pull around 5 ml of soapy bicarbonate solution into the corresponding syringe, and 5 ml of soapy water into the other. I'll invert the syringe, give it a few taps to make sure the discs are suspended in the solution, and then push the plunger until no air remains in the barrel. To create the vacuum, I'll hold my finger over the syringe opening and pull back on the plunger, holding the vacuum for 10 seconds and occasionally swirling the syringe during that time to make sure the discs are suspended. The syringe will leave a tiny imprint on your finger for a little while, but we must all make sacrifices for our science. After 10 seconds, I'll release the vacuum. The plunger should spring back. If it doesn't, that means the vacuum wasn't very good and you may need to try again or use a different syringe. If it works, the solution will replace the air spaces in the discs and the leaves should sink. Just be sure to release the vacuum gently so that the plunger doesn't spring back too aggressively. Sometimes even with a good vacuum, you'll need to repeat the procedure for the discs to sink. But if it doesn't work after 3 tries, the leaf discs will be pretty damaged. Best to start fresh with new discs and add more soap to your solution to help them sink better. And even after all that, you might only have 9 out of 10 discs sunk. But don't stress, just use that as your starting point for the experiment. Now that my discs have sunk, I'll pour them out into their corresponding cup.
Okay, who will explain what now happened? Nobody. Okay. So you see, we need these sort of discs to draw. So we want to draw them so they will be at the bottom, and when they will start photosynthesizing and generating some oxygen, they will go up, and we can basically see that. Otherwise, what will be the difference between the leaf that photosynthesizes and that does not? But the problem is that you cannot drown a leaf because it contains a lot of gas. So you see that spongy mesophyll and so on, so there is a lot of gas inside and you just cannot drown that in the water. So we are placing these parts of the leaves and we are putting them under the vacuum so that the gases from the inside, they will leave and will be replaced with the water. So what was now performed, that's some sort of replacement of that inner gases with the water. When they are replaced with the water, well, now they can sink to the bottom, and that's what we want.
Before I repeat this process with my other syringe, I'm going to cover the first cup with aluminum foil so that the discs stay dark and don't start photosynthesizing. Once all my discs are in their cups, it's time to put on my safety glasses and my gloves because I'm about to work with my bright light source and that can be shatter hazard. I'll put my cups under the light, take the aluminum foil off, turn the light on, and start my timer. The leaves in the with CO2 cup will start to photosynthesize and produce oxygen gas, allowing them to float to the surface. After a minute goes by, I'll record the total number of floating discs, then swirl the cup in case any discs have gotten stuck to the sides. I'll keep recording every minute until all of the discs are floating in my with CO2 cup. The discs in my without CO2 cup won't float because they can't photosynthesize without a source of carbon dioxide. So I actually waited about 15 minutes and one of my discs just didn't float, so.
Okay, so now what happened during the experiment? So what was the result? Can anyone explain? So we had 2 cups One was with baking soda, the other one was without. We had these sort of discs punched from the leaf. They all were ground at the beginning, so they started at the bottom of the cup. Then we turned on the light, waited for some time, and in one of the caps, these discs they started to float. So they floated to the surface. So what is the explanation of this effect? Magic. Yeah, almost. Yes, you're yes, you're right. So they started photosynthesizing. They have created oxygen. Oxygen does not dissolve in the water so good as CO2 does. Why? Because CO2 undergoes that reaction and you get that sort of carbonic acid, and oxygen does not. So you get oxygen in form of gas bubbles, and these gas bubbles they basically make that leaf to float. So that's all. Good. Now let's get further down. We are not interested in the whole leaf, but in some part of the leaf that basically performs the process of the photosynthesis. It's called chloroplast. So here is a schematic diagram and electron micrograph at high magnification that shows some some organized parts of the chloroplast. So here you see grana or granule thylakoids. Here they are in 3D. So everything will be going around these sort of structures. They are interconnected with some sort of stromal thylakoids, and stroma is basically the inner volume of the chloroplast. Of course, there are different sources of plastids that you can find in a leaf. Here is just some sort of an example, like starch-filled amyloplast, and this is a colorized scanning electron micrograph of a slice of a raw potato that shows cells filled with amyloplasts, and that's the organelles in which starch granules are stored. So basically, potato is a storage of starch. Chloroplasts are mostly grown, as I showed you, like these round spheres, but basically they can have different shapes. So here is just an example for you. On the left you see the photo of Spirogyra, and they have spiral chloroplasts that is good seeing. That's a freshwater algae, and here you see their chloroplasts. And chloroplasts of the higher plant cells that we'll be most interested in, they really have some more round shape, and here you can see them in this picture. So here are some sort of chloroplasts, and you can even see some internal structure. So they in this cell they are mostly on the cell periphery. Good. Here is once more anatomy of the chloroplast. So here we see the thylakoid vesicle. These are membranous, let's say, compartments, and we will consider what is happening there in just, I guess, in a few moments. So here are outer and inner membrane. Membranes, intermembrane space. A stack of thylakoids is called granum. Here you have thylakoid lumen. That's what's inside the thylakoid. We will need that because protons will be pumped inside, and we will get some sort of proton gradient, as we had when we in case of mitochondria, we have pumped our protons into the intermembrane space, and then we use that gradient to synthesize ATP. Here we will have a very similar process when we pump protons into the lumen, and then when we get this sort of gradient, we use ATP synthase that creates ATP for the plant. And here you see lamellae that basically interconnect all these sort of graina. Good. What else? One more video for you. These are living chloroplasts under the microscope. You see they are moving. This movement is not always random, but in many cases they can adjust in accordance to the light that they get. If you have too much light, they will try to move so that they don't get so much light. So they are trying to adjust themselves somehow to whatever conditions they are in. To the lighting conditions. So here you can see even some sort of internal structure. So if you look pretty closely, you see some sort of dots inside. So you might guess that these are graina, and that's basically, well, I don't think you can see something more with this sort of microscope, but under the electron microscope you can get much more details. Well, we just don't see them. They are there, but they are either too small or we don't see them at this point. So yeah. Good. What else? I wanted to tell you just a few words well, about the evolution of the chloroplasts. So chloroplasts, as far as we know, they are like mitochondria, and they contain their own DNA and the machinery to replicate and express it. However, they are not autonomous, so they also contain many proteins that are encoded by the nuclear DNA. So we suggest that probably they have just lost most of these proteins because the host cell provides everything they need, and there was no need for for synthesizing these sort of proteins. We now believe that in a manner analogous to the evolution of the mitochondria, chloroplasts are the result of endosymbiotic events in which a photosynthetic microorganism, most likely an ancestor of cyanobacterium, was engulfed by eukaryotic host. I guess that was after we have obtained mitochondria, so so yeah. That was the second event. What's more interesting that there are not one, but I guess at least two events when some sort of bacteria were engulfed, and we have different types of chloroplasts. Okay. What else? Some more photo. So these are chloroplasts from the outer layer of a green bell pepper. The dark specks inside are graina. You have seen that in videos, so Okay. Tunneling electron microscopy on the right. So here it is. Here once again we see all that graina, stroma, and so on and so forth. What's interesting, you see here a suspended starch grain. So they can basically synthesize and contain some sort of starch. Good. Here is a magnified part. That's a single granum from the spinac, and it's composed of 15 thylakoid membranes stacked on the top of each other. So here you see that in the micrograph. Good. What else? Just some sort of reminder that plastids may have different types and they can change. So here are some types shown for you and how they can interchange. Some are so when a proplastid is divided, it can transform into an amyloplast and let's say store some starch, or it can transform into the chloroplast, well, the most interesting organelle for us that performs carbon assimilation and starch synthesis and many other interesting syntheses. Depending on can it get some light or not, it can transform into something else like a thioplast or transform back into chloroplast, so that depends. So all types of plastids are bounded by a double membrane, and some have extensive internal membranes. Well, that's about the chloroplast. Internal membranes can be lost and resynthesized, so that depends. Proplastid in non-photosynthetic tissues such as root give rise to amyloplasts, which contain large quantities of starch, and all plant cells have plastids, and these organelles are the sites not only of photosynthesis but of other processes such as synthesis of essential amino acids, thiamine, pyridoxal phosphate, flavins, vitamins, so on and so forth. So here is an example, once again, what can happen to a chloroplast, that it can transform into pregranule plastid and back and so on. Good. One more tunneling electron microscope. So here is highlight and low light chloroplasts. So do you see the difference between these two chloroplasts? So what happened to the highlight and in the highlight conditions and in the low light conditions? Light dark bubbles. Well, it's not what I want you to focus on. Do you see what happened to graina? In which conditions do they have more these sort of disks? So more thylakoid disks in case of what, low light or high light? It's wider with low light. Yes. So when the light is low, you have more these sort of disks. The thing is that if you have highlight conditions, it's not so crucial for you to have so many disks because you have many photons, so you eventually capture some of them. So it's not a problem to capture a photon because you have many of them. So why would you create some large machinery? You can get away just with much smaller number of these sort of organelles, and that will be pretty okay. So why you waste some sort of energy for synthesizing all that membranes and so on and so forth? You have enough light, and eventually you will get your photons, despite some of them will just go through and so on. Moreover, plants don't even want to get too much light, well, due to some certain reasons like synthesizing of reactive oxygen species and so on. In the case of low light conditions, you may have some troubles of capturing these photons, and you will need to create more and more of these disks to enlarge your chances to get that photon that is coming to the leaf and use it for the photosynthesis. So that's the logic. Here's one more nice video. That's a photosynthesis under the microscope.
This is a plant leaf producing oxygen bubbles by the process of photosynthesis. This footage is sped up in order to show oxygen bubbles.
Okay, so what's this? What are these bubbles? Oxygen. Yes, absolutely correct. That's oxygen.
Bubbles growing. Photosynthesis is a process used by plants, algae, protists, and some bacteria to produce carbohydrates and oxygen from water, carbon dioxide, and sunlight. Organisms use photosynthesis to create food for themselves in the form of carbohydrates. This is done by an organelle in cells called chloroplasts. We can see them moving inside these plant cells. They are the green round structures seen here. Chloroplasts can move around in order to get more sunlight when there's little light, or move away from strong direct sunlight to avoid damage. Just like humans, too much sun can be bad. This process of chloroplasts moving to control light exposure is called photorelocation. Here we can see an oxygen bubble growing and the chloroplast in the plant cells moving around. We see a lot more oxygen bubbles than carbon dioxide bubbles for a few reasons. Carbon dioxide, or CO2, is more soluble than oxygen in water. This means carbon dioxide dissolves in water, similar to how salt dissolves in water. So when oxygen levels start building up in the water, these oxygen bubbles form more than carbon dioxide ones do. Carbon dioxide also reacts with water to produce carbonic acid, which is a completely different chemical. And carbon dioxide is also used up in photosynthesis. When plants photosynthesize, oxygen is somewhat of a waste product. They do need oxygen just like animals do, but they end up producing way more than they actually need. That is a good thing for all other living organisms that rely on them for oxygen. But that is also a bad thing for organisms that can be harmed by oxygen. These organisms, called obligate anaerobes, live without oxygen and can actually be killed by it. In fact, over 2.5 billion years ago, the Earth had very little oxygen in it. It was filled with anaerobic organisms until cyanobacteria came along. Cyanobacteria, which are photosynthetic bacteria, slowly produced oxygen over millions of years. The rising levels of oxygen slowly killed a large number of species on Earth and created a large extinction event. But this large extinction event helped set up the next stage of life, multicellular organisms and eventually animals. Photosynthesis is a very complicated but important process in biology. While all of the chemical processes of it can't be completely seen through a microscope, the oxygen produced at the end is, and when the footage is sped up, it produces very beautiful videos. So enjoy this biological marvel.
Okay, so that was the process of the photosynthesis under the microscope, and you see how nice that looks like. And really an interesting point that I'm not sure, but I guess I will show you some sort of processes. Plants also need, they also use this, or they use oxygen for certain processes, but they don't need it so much as they produce. So basically it's not a complete waste for them, because they use some oxygen, but they don't need it so much as we do, for example. And what's more interesting, there was really some sort of extinction event that was caused by the organisms that produce oxygen, because these oxygen was toxic for the previous for the anaerobic organisms. So when some organisms began to synthesize oxygen, they just killed a lot of different species. So yeah, if you look into books about the evolution, you will probably find something about this event. Good. here are once again some photos, and we get into the C3 photosynthesis. C4 will not differ very much, at least in the part of capturing light and generating something from that light, but when we get to the Calvin cycle and so on, we will see the difference there. Now, here is a typical C3 chloroplast. I guess you saw this image. Once again, they have a double membrane envelope and the internal system of membranes, thylakoids, that is suspended in the protein-rich fluid called stroma. And in a functional chloroplast, thylakoids form a closed, flattened velum. So this velum is closed, and that will be used to pump inside some protons and to get some proton gradient that is later used to synthesize ATP. Good. What else? Photosynthesis occurs in two distinct phases. The first one is called light reactions that use light energy to generate NADPH and ATP. The other ones are called dark reactions. That is extremely bad naming. Because when we read these names, they some sort of suggest that one part of the reactions go when we have light, say in a day, while the other part, I don't know, is performed at night. But that's not true. That's definitely not true. This is a very, very bad naming. So much better would be to call them, say, light-independent reactions. So these reactions are dependent on light because they use the energy of the photon to do something, while there are different reactions that do not use photons directly. They may use some sort of products from that part of the photosynthesis, but they do not use photons directly, so they don't need light in a sense that photons are not used up in these processes. But it doesn't mean that they should undergo just in a complete darkness. That's not true. So please remember that. You will see these namings in many books, and these are extremely bad namings. So that's nothing we can do. Nothing we can do in this case. Okay, so the light reactions, they occur in the thylakoid membrane and involve processes that resemble mitochondrial electron transport and oxidative phosphorylation. In photosynthetic prokaryotes, which lack chloroplast, the light reactions take place in the cell's plasma or inner membrane, or in highly invaginated structures derived from it, called chromatophores. So we will not get into that. I am mostly interested in leaves, but just for you to notice. Good. What else? The process of photophosphorylation resembles oxidative phosphorylation. So we will capture photon. This energy will be transferred to an electron. Electron will flow through a series of membrane carriers. So that should remind you on the electron transport chain of the oxidative phosphorylation that we had in mitochondria. So in some sort of philosophical sense, they are pretty similar. So we will have electrons moving through that carriers that will produce a proton gradient, pretty similar, and we will use that proton gradient to generate ATP. In oxidative phosphorylation, we remember that electron donor was NADH, and the ultimate electron acceptor was O2 that formed at the end H2O. Yeah, so here will be a little bit different. Good. In photophosphorylation, electrons flow in the opposite direction. So H2O, just water, is electron donor, and NADPH is formed. Water is a pretty poor donor of electrons, as you might guess, and its standard reduction potential is 0.8 volt, that compared to minus 0.3 volt for NADH. That was a good electron donor. So photosynthesis requires the input energy in the form of light to create a good electron donor and a good electron acceptor. And I guess I will show you how that happens. Electrons flow from the electron donor through a series of membrane-bound carriers, including cytochromes and kenones. We had pretty similar situation with mitochondria, and iron-sulfur proteins. We saw some sort of iron-sulfur proteins in the previous lecture, and I guess even in the electron transport chain. While protons are pumped across the membrane, and electron transfer and proton pumping are catalyzed by a membrane complex that is homologous in structure and function to complex 3 of the mitochondria, though they are pretty similar in their structure and their function. Maybe some sort of distant relatives, who knows. The electrochemical potential so produced is the driving force for the ATP synthesis from ADP and inorganic phosphate, catalyzed by a membrane-bound ATP synthase complex that is closely similar to that of mitochondria and bacteria. Good. So the part 1, we are harvesting some energy. So what we need to do, we need to capture some light, because light carries energy for us. Here is the example of the light spectrum. You see different wavelength, and of course we are interested in the visible light. I will show you what are exactly the energies of the photons that are used during the photosynthesis. But anyway, the spectrum of electromagnetic radiation and the energy of photons in the visible range can be measured, let's say, for 1 mole of photons. So anything countable can have 1 mole, yeah, because that's just 6 times 10 to the 23rd of some particles, in this case photons. The energy of a photon of visible light ranges from 150 kilojoule per Einstein. That's the naming of one Avogadro of photons. That's for the red light, 150, or 300 kilojoule per Einstein for violet light. So let's say quite different. Good. Here are the molecules we will have to deal with. So these are primary and secondary photopigments. Chlorophylls A and B, and bacteria chlorophyll. So here they are all shown. Well, shown is one of them, chlorophyll A, but here are some notions that will tell you how do you need to change this formula, say just a little bit, to get different form of chlorophyll. And there are certain other pigments that are used to harvest the light or to do something with that light. These are phycoerythrobilin and phycocyanabilin, so phycobilins. They are the antenna pigments in cyanobacteria and red algae. I will show you in a moment what are these antenna pigments and why are they used. So it's some sort helps to get the light, to harvest the light, and then that energy is carried to the reaction center where chlorophyll is performing the reaction of the photosynthesis. We'll get to that. Good, some lutein, and that is accessory pigment in plants, and so on. You can see here these sort of conjugated bonds. I hope you remember from our previous lectures what happens when we have lots and lots of conjugated bonds, and how that influences the energy levels in the molecule and fine-tunes them so that they are in the range of the difference between energies between HOMO and LUMO becomes in the reachable range for visible light. So photons will have enough photons of visible light will have enough energy to take electrons from the highest occupied molecular orbital and throw them into the lowest unoccupied molecular orbital. Here is some sort of absorption spectrum you see on the right. That's for different chemicals that we see here, so chlorophyll A and B and so on. So you can see them, and this black line, that's basically the spectrum of the sunlight that reaches the Earth. So if you take into account the real spectrum and what we get through the atmosphere and so on and so forth, if all the effects are taken into account, well, you can just measure that spectrum. You get something like this curve. Good. What this? These are some sort of primary pigments used by different organisms. Once again, I'm not going to get to go through all these tables. It's just some sort of reference for you to see that different chlorophylls may be used for may be used by different organisms, and they have different absorption peaks. Pretty nice strategy, because depending on the conditions you live in, it may have sense to somehow fine-tune the range in which you are absorbing the light. So if you are living somewhere in the water, you know that water absorbs light as well, so you have some shift in your spectrum, and you may just not get enough light in the part of the spectrum that is pretty good if you live above the water. Or you may compete with different species, and that different species, they are using some part of the spectrum, and that may be pretty reasonable to fine-tune the part of the spectrum you use to be out of the tune with that competing species, and get your exclusive ecological niche. Good. Here is an example of the biosynthesis of chlorophylls. You might guess, just by looking at the chlorophyll, that it pretty looks like a heme that we use for carrying oxygen. Of course, heme does not have that phytol tail. We had that. Let me find the formula. So here is the phytol side chain. Of course, heme does not have it, but the rest of the chlorophyll is pretty similar. And if you look at the process of the biosynthesis, you can see that even this process is pretty similar as well. So we can start with glutamic acid. We do certain reaction, we get to porphobilinogen. If you ever heard about bilirubin and all that sort of analysis that are performed in different clinics, that has to do with the products of heme metabolism. And basically here you see certain chemicals that may, by their names, remind you something along that lines. So here we go, and here is the part when we get protoporphyrin, and we can basically make a heme out of it. Or we can put here magnesium, yeah, because in a heme we use iron. So we can put iron in here and we get a heme, or we can put magnesium here and do some more and more reactions and get to the chlorophyll A. Or at some point we may change some parts of the molecule, let's say this and this one, and at the end of the day we can get chlorophyll B. So I'm not going through all of these reactions. I don't really think it's needed, but I just want you to know that more or less we have some general idea about the chemical pathway, how can we get chlorophyll, And along that way, we get certain chemicals that can be transformed into a heme. So if you will ever consider some pathway for I don't know, some pathways that are performed by the liver or some from other places, you will see these parts in the synthesis of heme and so on. Good. Now let's get to the capturing of the light. When the light comes, it is absorbed with the pigment molecule. What can happen to the light when it is already absorbed? So what can be the fates of that light? Well, if we captured that photon, that means we have transferred an electron from one orbital to the other. So that will be some sort of molecular orbital. This is some schematic view. Of course, it's not an atom, it's the large molecule. And when this electron is promoted, it may do, let's say, different things. It may dissipate its energy just, let's say, thermally. So transform that into sort of heat, and the photon will be wasted. Can be, of course. It can generate a new photon with the same parameters that the old one had, so we will get some sort of fluorescence. Pretty okay. It can transfer the energy, so this electron goes down, so from some sort of higher level it goes to the lower orbital, but in the expense of that energy, some different molecule gets this energy transferred. You can consider that as sort of generating a photon that is immediately captured by a nearby molecule. So you have one molecule, it was in an excited state, then it transferred back to its base state, generated a photon, but that photon was immediately captured by a nearby molecule that was transferred to an excited state. And in this sort of way, you have, let's say, transported that excitement from one molecule to another one. Or, of course, you can perform the photosynthesis. So you can transfer energy, or you can use this energy to do something useful. Good. What else did I wanted to say you? I wanted to say you something about carotenoids that are largely linear polyenes, such as lycopene and beta carotene, and that are components of all green plants and many photosynthetic bacteria, and are therefore the most common accessory pigments. They are largely responsible for the brilliant fall colors in deciduous trees, as well as the for orange color of carrots. So when the thing comes to the autumn, so in the fall, all the trees are thinking, well, it's time to get rid of our leaves, but there are pretty useful chemical components in there. For example, I don't know, magnesium ions in the chlorophyll and so on. So they are they start metabolizing that chemicals. Chlorophyll gets destroyed, and you start seeing the colors of all these auxiliary pigments, including well, this sort of beta carotene and lycopene. Good. Yeah, and even some plants, they additionally synthesize anthocyanins in the autumn. That has to do with the problem of reactive oxygen species. So they want to stop the process of capturing the light, because that generates reactive oxygen species, and they of course don't want that to happen. So they generate some additional pigments just to to handle the photons, and don't let them to get to the chlorophyll molecules and generate something they don't want. Good. What else? Here is an example of light harvesting complex. So here is the photo that came to this complex and was basically uptaken by one of the light harvesting pigments or antenna molecule. As I told you in the previous slide, this energy can be transferred to a nearby pigment molecule. That's performed in a random way, but if that interaction is pretty if that excitement is pretty long-living, you can basically wait long enough, and at some point it will get to the reaction center that will perform photosynthesis. Well, or namely use that energy to do something useful. So here are images that show you how that's all done. So these are shown antenna chlorophylls bound to protein. So here are carotenoids and accessory proteins. Here is the reaction center. We will get to that in a moment with more details. And here you see that light, that energy of the light is transferred to the reaction center, that where it will be used to generate something useful. Well, ATP for example. Good. Here are some videos that will probably remind you our first lecture, and their goal is to demonstrate some mechanical analogy to show you that sort of resonance energy transfer. So here is video. That's just a mechanical analog. Of course, we can't have some full analog because one is a quantum process, the other is just a mechanical process, but some similarities, I guess they are pretty enough. So here you see two penduli in this video, and do you notice anything about them? So what has happened? So we started by swinging just one pendulum. So let's look once more. What happened to the energy of the pendulum number 1 that we have, let's say, excited? Yes, they transfer energy to each other, absolutely correct. In the pretty similar way, an excited molecule can directly transfer its excitation energy to a nearby unexcited molecule with similar electronic properties. And this process occurs through interactions between the molecular orbitals of the participating molecules in a manner quite analogous to the interaction between coupled mechanically pendulums. Of course, they should have similar frequencies. So I remind you the lecture number 1. If you have similar frequencies, you can get some sort of interaction. So here we have it. So energy can be pumped from one molecule to another. And performing that sort of pumping, so here is one more example. So here you see when photon comes, its energy is transferred to the nearby molecules, and then, well, many things may happen. Basically, it can just go away in some cases, maybe. It can be lost in a sense that this energy will be dissipated into a heat, but it eventually, many photons will get to the reactive centers, and the reaction will be performed. So here is example of this exciton transfer. So you have this sort of excitation and electrons, with the help of the photon, are transferred from here to here. So you get an electron here, then this electron returns, but new electron gets up. Yeah, so you get another molecule excited, electron returns, but this gets excited, so on and so forth. But at some point, you get to the reaction center. It has a little bit lower energy than here, so it's energetically favorable to get to the reaction center. I mean, these electron will get down, but reaction center will be excited, and then reaction will be performed. Okay, so experiments have shown that most chlorophylls function together. Light, that is, they act as light harvesting antennas, so they do not perform some useful reactions, they just harvest light and pass them further. These antenna chlorophylls pass the energy of an absorbed photon by exciton transfer from molecule to molecule until the excitation reaches a reaction center. There, the excitation is trapped because reaction center chlorophylls, also chemically identical to antenna chlorophylls, they have slightly lower excited state energies because of their different environments. I guess we will get to that in some of the future lectures. If you have even absolutely identical molecules, but in something living in some biological system, they will probably exist near to some proteins or parts of certain proteins. And these proteins, depending on their structure, they change the environment of the molecule, and that may change some characteristics of the molecule. For example, rhodopsin in our eye, it's absolutely the same in all the rods and cones and whatsoever. But it is inside a protein, and these proteins are a bit different. And these different proteins, they make rhodopsin to shift its energy levels, and that's why you have, for example, some cones that absorb blue light and other cones that absorb red light. Because different environment influences the molecule, and this influence may change something like energy levels of the molecule. This is a pretty similar case for the chlorophyll. So you have some molecules outside of that thing, outside of the reaction center, and they have one absorption spectrum, while you have different chlorophyll in the reaction center, and it will have a little bit different absorption spectrum just because of the environment. Good. Here we are getting to the reactions themselves. So what is happening in the with the light that we have acquired, and how do we perform the process of the photosynthesis so that we can get something useful, not just transfer electrons from here to there, but create some useful chemicals. Photosynthesis is more complicated in green plants than in photosynthetic bacteria, because in green plants photosynthesis depends on the interplay of two kinds of membrane-bound light-sensitive complexes, photosystem 1 and photosystem 2. I guess we will consider them both. Here is a simplified scheme with defined wavelength of the light that are absorbed by these different photosystems. You see that one of them can be used is used to generate NADPH, while the other one creates a proton gradient and just passes electrons to the photosystem 1 at the end of the day. I guess we will get to some more detailed schemes in a moment. Okay, what that? That's how they look like with respect to all the organelles we have considered. So photosystem 1 occurs mainly in the unstacked stroma lamellae in contact with the stroma where it has access to NADP. Photosystem 2 is located almost exclusively between the closely stacked grana out of direct contact with the stroma. And cytochrome B6F is uniformly distributed throughout the membrane. So here you see the comparison between oxidative phosphorylation. So here we had what? Inside was the matrix, so this was the matrix. Here we had intermembrane space, so there was one more membrane on the outside, so one more membrane, and that was a mitochondria. Here we have pumped our protons into the intermembrane space, from the matrix into the intermembrane space. We got some gradient, some proton gradient, and this proton gradient was used to generate some ATP from ADP. Now let's check photosynthesis. Here inside is thylakoid space, outside is stroma, one more membrane outside here, and now we are pumping protons inside the thylakoid space. We are creating gradient, they are going outside and generating some ATP. Here is one more view. So here you see how these ATP synthetases are situated, and here you see all these sort of proteins, how that looks like. So these are, this is grana. Here you see inside, that's a thylakoid space. That should be a continuous volume, enclosed. Of course it should be enclosed because we want to keep proton gradient across this membrane. And interesting question for you before before we move further. If we consider oxidative phosphorylation in comparison with the photosynthesis, I want you to focus on the ATP synthase and how the proton gradient is created. So probably we can think of thylakoid space as some sort of homologue of a matrix in mitochondria. We can probably think about stroma as a intermembrane space in the mitochondria. So how is the ATP synthase situated in these two cases? Similar or different? And what's different? Where are protons pumped in both cases to create the gradient? Yeah. Okay, let's look at this way. So pretty schematical. So we have two membranes in case of both oxidative phosphorylation and in case of photosynthesis. Good. If this is a mitochondria, what do? We pump protons into the intermembrane space. So here we have some plus charge. Well, let's say these are protons. And then we have ATP synthase that synthesizes ATP by letting these protons in. Now the case of the photosynthesis. I have two membranes, but now I'm pumping protons in here. So these are my protons. And here I have my ATP synthase that generates ATP by letting protons out. Let me, I guess, go to the end. Because for the homework you will have to consider an experiment that is shown here on the right. So here you have a thylakoid membrane, and there is an experiment that basically putting this membrane in different solutions with different pH, you are basically inducing the same pH inside. What is pH? That's how many protons, how many free protons you have in there. And here you see that putting that in a higher pH, so when there is a lower concentration of protons on the outside than on the inside, they are starting to going out and they are generating some ATP for you. That's a real experiment. And that has to do with the with that scheme that I showed you where we are. That I showed you here. Okay. Okay, I guess I'm getting we're almost out of time. Let me check just for a moment. Maybe I'll get to all these details next time. Just few words at the end to somehow finish this part. So two photosystems. I will consider how do they work in more details tomorrow. Just few words. So photosystem 2 is the pheophytin quinone type of system, like a single photosystem of purple bacteria that contains roughly equal amounts of chlorophylls A and B. Excitation of the P680 special pair in its reaction center drives electrons through the cytochrome B6F complex with non-committant movement of protons across thylakoid membrane. So we are basically pumping protons out. So where do we have a nice scheme? So here we have it. So here is that cytochrome. We are pumping some protons out. Photosystem 1 is structurally and functionally related to the type 1 reaction center of green sulfur bacteria, so we even know something about their evolution. It has a P700 reaction center and the high ratio of chlorophyll A to chlorophyll B. The excited P700 passes electrons through a chain of carriers. I guess we will consider all that carriers tomorrow. to the iron sulfur protein ferredoxin, then to the NADP plus producing NADPH. So we already get something useful out of it. The thylakoid membranes of a single spinning chloroplast may have hundreds of each kind photosystem. And these two reaction centers in plants act in tandem to catalyze the light-driven movement of electrons from water to NADP+. Electrons are carried between the two photosystems by the soluble protein plastocyanin. We had something pretty similar when we considered phosphorylative oxidation. To replace the electrons that move from photosystem 2 through photosystem 1 to NADP+, cyanobacteria and plants oxidize water, and green sulfur bacteria oxidize H2S. Good. Here are some nice pictures with an X-ray crystal structure of photosystem 1 in spinac, and here is X-ray of photosystem 2 in the marine cyanobacterium. Details we will get tomorrow. I guess here are some nice 3D views for you, so you might want to check that. I'm not sure. They will load correctly. Okay, while everything is freezing and loading, maybe we have some questions or comments so far. OpenGL does not work because it wants this thing. Good. Nope. Yeah, that's a typical problem with the browser, that it does not want always draw all that 3D structures. Maybe you will have some similar problems, so this is the way to fix that, at least in some cases. So to set use graphics acceleration when available. That breaks certain different things, but at least this one starts working, and then you can turn that off and get that other things working, but this broken. Yes, that's the way modern software works. Good. I guess I'll show you this 3D, and that will be, and we call it a day. Okay, loading. Yep. Okay, I guess. Here we are. So here is the 3D of the, I guess, photosystem 1. So I guess you can play with that and maybe see something interesting. And tomorrow we will consider some more details about the photosynthesis. Good. So if we have no other comments and questions, I guess it's a good place to stop. So thank you for your attention. Have a nice evening and see you tomorrow.